Darwin's second postulate is that in order for a trait to be acted upon by natural selection, that trait has to be, at least to some extent, heritable. It has to be passed on from parents to offspring. Why is this so critical to evolution by natural selection? Well, imagine that I have a beneficial trait that allows me to reproduce more successfully than other individuals. In order for that trait to increase in frequency in the population, I need to be able to pass that trait on to my offspring. There has to be some probability that my offspring will also have that trait. Otherwise, there's no way for it to increase in frequency in the population in the next generation, which is the definition of evolution by natural selection. And so what do I mean by to some extent heritable? So remember that when we talked about blending inheritance, we said that more complicated, continuously varying traits such as body height in humans can give the appearance of blending inheritance because of fairly complicated inheritance patterns. So we know inheritance is discrete, but gene expression can be complicated. So it can be controlled by multiple genes. There can be an effect of the environment, and the genotype and the environment can also interact in complicated ways that make it difficult to understand the heritability of certain traits. So in this web lecture, we're going to focus mostly on being able to distinguish between how much of phenotypic variation is due to variation in genes compared to how much of it is due to variation in the environment. And to start to think about this, we need to bring in this idea of phenotypic plasticity. That is basically the effect of the environment on phenotype. So this should be fairly intuitive to most of you. It's entirely possible that these two mice are identical twins. They have identical genotypes, possibly. And it's not very surprising to us that one of them is fat because it sits on the couch all day and eats potato chips and drinks beer. The other one is thin because it spends its day walking on the treadmill and apparently eating nothing. So this is not surprising to us, okay? We understand that the things we do and the environment that we're in have an effect on our phenotype. So if we're going to define phenotypic plasticity, that would be the ability of a single genotype to produce more than one phenotype when exposed to different environments. And we can measure this by looking at specific genotypes, their trait values, and the environment that they're in. So in this fairly abstract case, we can think of a trait such as, say, height of a plant in some environmental gradient. So let's say this side close to the origin is maybe hotter temperatures. Maybe we're going up the slope of a mountainside to colder temperatures on this end. And we take identical genotypes and plant them across this environmental gradient and measure the trait value that it achieves at adulthood, in this case height. So in this first case, if each of these lines represents a single genotype, so the red one is one genotype, the pinky purpley one is another genotype, the blue line is another genotype, we can see in this case there's no effective environment whatsoever. The plants get to be the same height for each genotype, no matter where in the environment they're grown, which part of this environmental gradient they're grown in. The pinky purpley one, again, a flat line, it grows to the same height no matter where it's grown, same with the blue. But there's a very strong effective genotype, right? These red ones tend to be the tallest, the blue one's the shortest, and the pinky purpley one's kind of in the middle. So no phenotypic plasticity in this case, but a strong effect of genotype. In this middle case, we do see that there is some phenotypic plasticity. The single genotypes are going to reach different heights depending on what environment they're grown in. So for all these cases, they grow the tallest in this warmer environment and not so tall as we get colder. This effect of the environment is not the same for all genotypes. So each of these three genotypes reacts to this environmental gradient slightly differently. This one has a very strong effect, indicated by the steep slope. This one, the blue one, not so strong effect, indicated by the more shallow slope. And these phenotypically plastic responses can actually become very, very complicated. So we can see in this last graph here that the effect of the environment depends entirely on the genotype. So this red genotype grows tallest in the warm environment and not so tall in the cold environment. This pinky purpley one grows tallest in the cold environment, but not so tall in the warm environment. And this blue one has very little response to the environment for this genotype. And one possible explanation for this is that the red and the purple one are locally adapted, and they were actually taken from the different kinds of environments. So maybe this one, the red one, is native to the warmer environment, and so it grows the tallest when it's in the environment that it's adapted to. So local adaptation doesn't do so well in the environment that it's not adapted to. Similarly, this purpley pinky one might have been taken from a colder environment, so this is its native environment. It grows best in its native environment and does not do so well in a warmer. And so this is called a genotype by environment interaction. How the environment affects the trait value depends very strongly on what genotype the individual has. And each of these colored lines is known as a reaction norm. So this is how the genotype reacts differently depending on the environment that it's in. And this is how we can measure phenotypic plasticity. So this is a very abstract version of this. We can actually demonstrate this in field studies. So these were done by a group of Stanford biologists on a wildflower, a native wildflower called Achillea. And the Achillea plant can be grown from cuttings. So that means you can just take a little cutting from a plant out in the wild and grow an entire plant from it. So for each of these plants that they found out in the wild, they took 2 cuttings. So each of those cuttings are going to generate an individual that has an identical genotype. They're genetically identical. So both of these labeled A are genetically identical. This is genotype A, genotype B. Both of these 2 plants are genetically identical, and so on down the line. So they took these pairs of genetically identical plants and planted one of them at a field site called Mather Field Station. They took the other member of each pair and planted them at Stanford's experimental garden. So they were all planted very close to each other at each of these sites so that each genotype experienced an identical environment at each of these locations. So same amount of sunlight, same amount of water, same kind of soil, everything identical in their environment, but they're in 2 different environments, one at the field site and one at Stanford. So the first thing to notice is that they reached different average heights when grown in different locations. So the ones that grew in Stanford all grew taller than the ones that grew at Mather Field Station. So this is the effect of the environment. You'll also notice, though, that within each environment there's still variability in plant height. So even though the environment was identical for all of these plants, the genotype that it has also had an effect on the phenotypic variability, and the same is true in the Stanford location. So there's an effect of both environment, shown by the difference in height between this whole group and this whole group, and there's an effect of genotype, which is shown in the variability within each group between the different genotypes. So this demonstrates this idea of phenotypic plasticity and the idea that although a trait has a genetic basis and there's variability in that trait, that's not going to completely determine the phenotype you get. Both genes and environment are going to play a part in the ultimate phenotype. We took a look at this in our hypothetical example that we did in class of breeding out dog breath. In this scenario, we were breeding just the dogs with the most minty fresh breath, and our hope was in the next generation, when we bred those dogs, we would have a shift in the mean breath value from more dog breath to mintier breath. But what we found, in fact, was that in the actual offspring, the mean breath phenotype was exactly identical to the mean breath phenotype in the parental generation. And we said that this was probably because dog breath is not a heritable trait. And so we can see that heritability is critical for selection to occur. We can only get this shift in phenotype from generation to generation. Okay, this is evolution by selection, artificial selection in this case. And we know that only genetically based variability is heritable. This is how we inherit things, through our genes. So how can we determine whether variability in dog breath is due to genes or proximity to the mint patch or some combination of the two? So let's very carefully define what we mean by heritability. What we want to know is how much of the variability in phenotype is due to variability in genotype. That's the only part of this variability that's going to be passed on to offspring, that's going to cause offspring to tend to resemble their parents. And we can put this in mathematical terms to try to understand it a little bit more clearly. So VP is the total phenotypic variance in the population. This total phenotypic variance is due to two things. One is variance due to genetic differences, and the other is variance due to environmental differences. We don't know of any other causes for phenotypic variation. It's a combined effect of genes and environment. And so this is what we know as broad sense heritability. So what we want to know is the proportion of the phenotypic variance explained by genetic differences between individuals. So if we give broad sense heritability the variable H, we want to know what proportion of this phenotypic variance is due to genetic variance. So it's going to be VG divided by VP. We also saw on the last slide that VP is equal to VG plus VE. So we can say that broad sense heritability is genetic variance divided by the sum of genetic variance plus environmental variance. I want you to keep in mind, though, that this is really an abstraction. This is just a conceptual model to help us clearly define these variables and what it is that we're looking for. VG and VE are not something that you can just go out and measure to be able to calculate broad sense heritability. So this is not a formula for calculating heritability. This is just defining our terms in a mathematical way. But not all genetic variability has the same effect on the response to selection that we're looking for, that shift in average values from one generation to the next. So as you'll recall from your genetics class, we have some cases in which different alleles have a dominance and recessive relationship. So we call this dominance genetic variation. And in this case, because the homozygote and the heterozygote have the same identical phenotype, going out and measuring the phenotypes in the population and the variance in the phenotypes is not going to give you an accurate measure of the variance in the genotype, right? Because all of the individuals with the dominant phenotype are a combination of homozygotes and heterozygotes. You have no idea how many of which you have. So it's not giving you a good understanding of the amount of genetic variance there is in that population. We also have something called additive genetic variation. In your genetics class, you probably learned this as incomplete dominance. So in incomplete dominance, remember that the heterozygote has an intermediate phenotype between the two homozygotes. So that means that there's this one-to-one correlation between genotype and phenotype. Each genotype has its own distinct phenotype. So you can go and measure the phenotypes in the population and have that give you a good estimate of the actual genetic variability in that population. To get a good idea of how closely offspring will resemble parents, that relationship between phenotypic variation and genetic variation, we use only additive genetic variation. To look at this a little bit differently, let's imagine a situation where there's a gene with two alleles, A1 and A2. And in this first case, we're going to say no dominance. Okay, so this is what we called before an incomplete dominance situation. And we're going to assign phenotype values to each of these genotypes. So the A2 homozygote, we're going to say has a phenotype value of 2. We can think of this as A1 being a loss of function allele that doesn't produce a functional protein. A2 is a functional protein. You've got 2 copies of it. You get twice the protein product. In the case of the heterozygote, you get sort of a half dose of the protein product, and we're going to give it a phenotype value of 1.5. And let's say in the homozygote, loss of function, we have a phenotype value of 1. That's what you get without any of this A2 protein product. So if we look at these phenotype values on a histogram showing the fraction of the population with each of these phenotypes, if you'll remember back to your Punnett squares, if you have 2 alleles of a gene, about half of the population will be heterozygotes, about a quarter each of each homozygote, and this is what your population is going to look like. So now if we plot genotype in terms of the number of A2 alleles. So these ones have 2 copies of A2, this one has 1 copy, this one has 0 copies. This phenotype correlates, this is the phenotype value on the y-axis, the phenotype correlates very nicely with the genotype. We can plot a line through those points and each point falls exactly on the line. We've got a very strong correlation between genotype and phenotype. So that means that there's going to be a very strong tendency for offspring to resemble their parents because each genotype has its distinct phenotype. Another way of thinking about that is that the variation in genotype is exactly reflected in the variation in phenotype. There's a 1-to-1 correlation. On the other hand, if you've got complete dominance, now our phenotype values are 2 for the A2 homozygote, also 2 for the heterozygote, because remember, complete dominance means that the phenotype of the heterozygote is identical to the phenotype of the homozygote. And then again, we've got a phenotype value of 1 for the A1 homozygotes. In this case, if we look at the fraction of the population with each phenotype, we've got 75% with 2, because that's a combination of both the heterozygotes and the A2 homozygotes. And then again, 25% of the A1 homozygotes with a phenotype value of 1. So now if we plot these genotypes in terms of the number of A2 alleles against phenotype value, we do not have a nice straight line that we can put between these that all the points are going to fall upon. There's not this 1-to-1 correlation between genotype and phenotype that's going to make this a very predictable tendency for offspring to resemble parents. So this gives us another way to define heritability. So remember, what we want to get at is that portion of heritability that is going to be important to this response to natural selection, that shift in phenotype value from one generation to the next. So now, because we know that there's this direct correlation only when you use additive genetic variation, what we want to know is the proportion of phenotypic variation that's explained just by the additive genetic variation. This is the part that causes offspring to resemble parents. So we're going to give this a new variable, and for historical reasons, this variable is called h squared. Please note that there's nothing that's actually squared here. There's no taking the square root of this and getting h. The number that you get for narrow sense heritability, the variable that you use for that is h squared. So h squared now is going to be, again, the proportion of the total phenotypic variability that can be accounted for by additive genetic variance. So additive genetic variance divided by total phenotypic variance. Again, we're going to split out phenotypic variance into genetic and environmental, and then we've also split out genetic into additive plus dominance genetic variance. So this additive plus dominance genetic variance together makes the V sub G that we saw in the broad sense heritability equation, and then V sub G plus VE is going to equal the total phenotypic variance. So again, this is sort of a conceptual construction to help us define our variables. These things are not things that we can just go out and measure directly. Additive genetic variance, dominance genetic variance, and environmental variance. But we do have a way of actually measuring this. Because this is the variance that causes offspring to resemble parents, we can measure this just by comparing offspring and parents. We make a plot like this comparing parent values to offspring values. So let's go through this graph point by point here. So each point on the graph represents the average value for the two parents, mother's value plus father's value divided by 2, plotted on the x-axis, and then the mean trait value for all of the offspring of those two parents on the y-axis. Okay, so this point is one set of parents and all their offspring represented. So what we would expect is that for parents with really bad stinky dog breath to give rise to offspring with really bad stinky dog breath, and parents with nice minty fresh breath to give rise to offspring who also have nice minty fresh breath down here. To get a measure of the narrow sense heritability, we basically just do a linear regression, get a best fit line through this cloud of points, and the slope of that line is equal to the narrow sense heritability. So narrow sense heritability, h squared, can vary between 0 and 1 and take any value in between. So this tells us something about the relative effects of genes and environment on producing phenotypic variability. So in this case, if we take that slope, it's approximately 0.5. So let's look at what each of these ranges of value mean in terms of how we can interpret these graphs. Let's start here at the bottom with a heritability of 1. So again, as I said before, this is, in this case, the axes are tall versus short, again, but this is mid-parent versus mid-offspring. Tall parents give rise to tall offspring. Short parents give rise to short offspring. There's a very reliable correlation. So what this means is that most of the phenotypic variation in height in this population is due to genetic variance. This is exactly the same set of points that we looked at over here. This is a heritability of about 0.5. There's some tendency for tall parents to give rise to tall offspring and short parents to give rise to short offspring, but there's quite a bit of scatter around this, and the slope that you get with the best fit line is about sort of halfway in between. There's sort of equal contributions of genetic variance and environmental variance that contribute to the phenotypic variance in that trait. In this top case, when we do our best fit line, we get a slope of 0. So that means there's no tendency whatsoever for offspring to resemble their parents. And we can see this clearly by showing that the very shortest parents give rise to kind of average height offspring. The very tallest parents give rise to kind of average height offspring. The tallest offspring came from average height parents. The shortest offspring came from average height parents. It really doesn't matter how tall your parents were in determining how tall you're going to be. So this measure of narrow sense heritability, this tendency for offspring to resemble their parents, gives you this very, very important piece of Darwin's postulates that give rise to natural selection. How much of this variability in the population is actually going to result in a shift in trait values in the next generation? In other words, evolution by natural selection.